presenter. So we have Dr. Michael J. McTavish, and they're a research associate with the Smith Forest Health Lab at the University of Toronto. He has a research background in the fields of ecological restoration, invasion science, and the biological control of weeds. His current research focuses on a biological control of introduced Phragmites and garlic mustard, and the ecology of non-native earthworms. So I will let them take it away, and I'll stop sharing.
Great, thank you very much. I'm just going to pull up these slides here, make sure that's all good. Okay, that coming through all right?
I think there's just a slow delay. Yep, that looks good. And it's in presentation mode, so you're good to go.
Okay. Perfect, thank you. Well, thank you for joining us today, everyone. It's great to be back and speaking with this group in 2022 as well. And there's been a lot of progress since that time, so excited to share some of that. I also want to extend a thank you to Teresa as well for coordinating and facilitating all of this. So we're going to be providing a bit of an update here on the biological control program for Phragmites as it exists still just in Ontario, Canada right now. I know there's a lot of interest in this, including across the border, and we'll touch on some of that, and sort of documenting at a fairly high level how this has moved from something of a research stage in the initial years to more of the integration sort of orientation of it that it's in now, although of course still supported by research throughout. With this group, I think we're all quite familiar with introduced Phragmites, why it's a problem and why it's a challenge, broadly considered one of the worst weeds in North America. There are a lot of excellent success stories out there in terms of control that does work, but there are many limitations with this as well, and as many other cases where it just isn't getting the job done. Individual efforts may be small scale or short term and need a lot of reapplication. As you know, it can be very labor intensive and often extremely expensive, and that can really be one of the main limiting factors. Any control has to grapple with just how widespread Phragmites invasion is. And of course, this is a very aggressive, persistent plant, so the threat of regrowth or reinvasion or other forms of site disturbance also need to be factored in. So what we're doing here, as we always say, is we're not trying to replace the tools that we already have. We're trying to add instead another tool to the toolbox. And in that case, the tool we work with is biological control or biocontrol. In the most broad sense, biocontrol uses living organisms to gradually suppress a pest at the large scale over the long term. So we're trying to address some of those shortcomings that we have in other tools that sometimes work very well, but aren't as large scale, aren't as long term. Biocontrol works because of is trying to establish this kind of natural balance that exists in the home range. So in their native habitats where these species aren't weeds, species are generally kept in check by natural enemies. So these might be diseases or in this case, herbivores that again, just keep those populations from effectively getting out of control. When they invade as a non-native species, they can escape and leave those natural enemies behind. So without that counterbalance, they can become more competitive, more dominant, effectively acting now as invasive populations in that new range. And so at the base level, biocontrol is really just about reestablishing that natural ecological balance that exists in the home range, now in the invaded range, by bringing over one or more natural enemies, very host-specific natural enemies, that can suppress that population at the long term. So it's not removing it, but it's present at a lower density, effectively acting much more like a native species, where it's just not causing the same kinds of problems. It's really important for biocontrol to understand these goals, what we're trying to achieve and what we're not trying to achieve, what we're not expecting. And the goal here is, again, gradual long-term suppression, so not eradication. So you can imagine a sort of hypothetical scenario where we have a pest like Phragmites that exists at a very dominant, problematic, damaging level. Biocontrol agents, one or more, are introduced at fairly small sizes. So these are natural organisms that reproduce on their own, so we can take advantage of that. Over time, and time here is really measured in years, and that's an important aspect of all of this, is that these are longer term programs that take 5, 10, 15 years to really sort of see demonstrable impacts. But over that time frame, we start to see spread of biocontrol agents and decreases in pest populations. And finally, where we want to end at long term is agents and pest populations present, but at very, very low density, sort of cycling and fluctuating back and forth, again, much like any sort of native vegetation and its herbivores would sort of fluctuate at. So biocontrol I've already outlined has some advantages associated with it. We'll get more into this, but only agents that are demonstrated to be highly host specific to the target are used, and so it can have very low environmental impact. It is a very targeted tool compared to some of the other things that we might be using, and very safe, very focused for that reason. The fact that it is actually very gradual and low disturbance, that this takes many, many years, is actually good in the sense of reducing overall site disturbance and by reducing the risk of new or reinvasion. So we're not leaving behind a large empty niche in our ecosystem. We're sort of gradually suppressing it, and ideally we're seeing replacement, which we may foster with restoration of more desirable native biodiversity. Because these are living organisms, they are self-sustaining and self-dispersing to a certain extent. And for that reason, this tool tends to be very cost-effective, can operate at very large scales, and is, again, a long-term solution that once it's in place, will continue to be in place without reapplication. So biocontrol is often sort of what we turn to for longer-term management of large-scale invasions that are just too costly to manage otherwise. If you're familiar with the invasion curve as depicted here, essentially over time, as an invasive species spreads and occupies a much larger area, the cost associated with management at that scale becomes larger and larger, eventually reaching a point where certain controls just become sort of impractical to use merely by expense. And so that's where we start to look at tools like biological control. So if we look at the Introduce program, the program rather for Introduce Phragmites specifically, and sort of go through the story of how that came to be here. The biocontrol agents we're using at this point are 2 European moths, so they're from the native range in Europe, and they have stem-boring larvae, so you can see them here as caterpillars and moths. This is Archanara neurica and Lenisa geminipuncta. They're fairly small, drab caterpillars and moths, relatively similar in many regards, but we do have 2 agents for this system. Again, the life histories differ a little bit in terms of timing, but sort of more similar than different. The larvae hatch in the spring, and they'll feed in the shoots for about 4 to 6 weeks. That's the period of time where you're getting the actual damage to the plant. Once they've grown, they then pupate in the stems for about a month. They emerge as adults that'll fly for 1 to 2 weeks, ideally mate, and then lay about 150 eggs per female underneath the leaf sheaths of the plants. So they'll oviposit under the leaf sheath up against the stem, and as the plant senesces through the fall and the winter, the eggs stay there sheltered on that standing dead biomass until the following spring, when they then hatch. The damage comes from the larvae, as I noted. So these are stem miners. You can see a larvae of Lenisa geminipuncta there, chewing through that interior tissue of the stem. They'll switch between 3 to 4 stems as they develop, chewing their way out and moving to another stem. This leaves behind very characteristic boreholes, as you can see pointed out here, and generally death of the tissue above that feeding point. With younger stems, those stems just kind of can't support that amount of damage, and they're usually killed outright. Older stems can sometimes survive this if they're large and robust enough, but their growth is effectively halted at that point because they can't continue to grow past that damage. And so overall, because of this feeding, so death and stunting of stems, the larvae reduced Phragmites stem density, the height, and the probability of stems forming panicles, or that seed head. So collectively, this makes Phragmites less dominant on the landscape, less competitive, less reproductive output. Again, with this ideal long-term goal of increased biodiversity and function. So importantly, Phragmites is still present as a part of this community, but not in the same dominant sort of problem-causing way that we're seeing currently. So it was pointed out, so I did a version of this webinar in 2022, and at that time I pointed this out, and it is just as prescient, maybe even more so now, to really sort of highlight that introduced Phragmites is very much an international problem. It doesn't respect borders, as we well know. And so development of this program has really been an international multi-stakeholder solution. And we'll get into a bit of a timeline here, but it's at this point now over 2 decades old. We have a lot of key personnel based in Canada, but also in Europe, and many partners and collaborators in the US who we continue to work with as well. So really, it takes a lot of people, and it takes a lot of institutions in different places, which again, apparently is an important thing that we need to remember right now. This is a bit of an updated list of some of the key partners that we're working with currently, these sort of large-scale collaborative networks that make this work possible. Our core team is relatively small, with a relatively small amount of funding, but we benefit from partners, financial partners, research partners, land owners, everything from environmental non-government organizations to conservation authorities, private friends of groups, municipalities, universities, and so on. So right now, a large chunk of our funding in particular comes from Ducks Unlimited Canada, but we have close partnerships with a variety of other groups as well. And this is really what makes this work even possible in the first place. So I'm going to sort of show a program timeline in a couple of different ways. Here's a bit of a just high-level overview. The program really started in 1998. So again, these things take time. This is when the initial proposals were put forth, and there was an exploration for insects that would be suitable candidates as biocontrol agents. These were identified, and testing began in the mid-2000s of the species that we now are still using. So a lot of time goes into ensuring that host specificity, and we'll look at that again here in a second. This all culminated in a petition to release these two agents in Canada and the US. These are connected petitioning process. The Canadian petition was approved by the Canadian Food Inspection Agency in 2019, and that's when releases began. The US petition remains under evaluation. The agents have been recommended for release, but there are some additional sort of steps, and it's a process that sometimes take a little bit longer. So we're still hoping for that to come through sooner rather than later, but that's where it is at currently. So if we look at that same timeline, more in terms of our work on this and sort of the broad chunks of time working on this, you can kind of imagine an initial phase leading up to that release permit. Again, focus on identifying the agents and getting that permitting in place. So this was, again, that international team across various borders. And the focus here is really on identifying suitable agents, with that suitability being based primarily on specificity. Are they able to feed and develop only on the target weed? And this requires very extensive testing in the field and under quarantine conditions, looking at introduced Phragmites, but also taxonomically related species and other vegetation as well, including crops and ornamentals, economically significant species, and so on. So once it was established that this specificity was in place, we saw the Canadian release permit being approved by the Canadian Food Inspection Agency in 2019. That's when I got involved with this work as well through the University of Toronto. Broadly, the second phase was really focused on, okay, we're allowed to release these agents now. How do we do that? Effectively developing a set of operational protocols for how to rear the agents, release them onto the landscape, and then monitor their performance and impacts. Very, very quickly to look through these steps here, you need insects to be able to release. Fortunately, we are able to rear through the entire life cycle in the lab to produce more insects. So often we're starting at the top there with eggs. We can hatch these out into larvae that are fed either cut stems or an artificial insect diet, can produce pupae, produce moths produce more eggs. So we can go through the whole life cycle, and we can also take any one of these life stages and pull them out of the rearing cycle for release. So you have to actually get them onto the landscape. In general, you can't just sort of take them outside and dump them. You do need to be a little more refined than that. We've experimented with various methods and have successfully used releases of all the different life stages here. So eggs, larvae, pupae, and moths. They tend to have different pros and cons associated with each of them. Right now, we're focusing on best practices using a combination of eggs and larvae. So the eggs go into these screened protective cups before the eggs hatch, and we stake those out in the field in the spring. The larvae will hatch out, crawl out of the cup, and go find a Phragmites stem to start chowing down. The other approach is that we can let them hatch in the lab and then inoculate by hand larvae into cut Phragmites stems about 30 centimeters or tall here. And you can see them assembled in blocks of floral foam to keep them hydrated, and then put those blocks out into the field. They'll feed in there for about 1 to 2 weeks, And then when they're done with that stem and ready to switch to another, they chew their way out and switch to another stem. You can see here a comparison of the amount of feeding damage that we see associated with egg and larval releases. And you'll note that it is higher for larvae. So we know this is a more effective method overall per amount of number of insects released, but it also takes a lot more work. So in general, what we're using is a combination of the efficiency of egg cups and the efficacy of stem larvae across our various releases now. Once they're in the field, you also need to be able to monitor them to see if that population is persisting, if it's growing, if it's overwintering, effectively, is it establishing on the landscape? These are internal feeders with very drab, sort of relatively inactive moths. And so often we're looking for indirect monitoring evidence here. So some of the things we'll look for would be those characteristic boreholes, the wilting of the stem that results from the damage. Less often, but we can also look for plant responses to that damage, like secondary branching. They'll put out secondary shoots to try and compensate for the loss of that main stem there. You can also see on the far right a pupal window. So this would be what's left behind after an adult moth has emerged from the stem. So we can quantify all of these in the field. We've developed standardized protocols that we can use year after year to detect that agent feeding damage. That's the main way we're monitoring the populations themselves. And we have to do that a little bit differently in the first year and the second year. So on the far left, you can see here, when we're putting out eggs or larvae, we kind of know roughly where they'll be because they only move a couple of meters in that first year. So we can really focus our monitoring on a pretty specific area. But once they've had a chance to fly around as moths and choose their own oviposition sites, and they're often better at choosing where to go than we are at choosing for them, you need a bit of a different protocol. So we have sort of a rapid version of assessment for feeding damage that we will deploy using these sort of monitoring interval plots on quadrats that we run throughout the whole Phragmites patch. So with that in hand, really what we've started in just the last couple of years to focus on now is more of a scaling up. So we have those core protocols, the agents are here, we know how to work with them, and basically we need to get them onto the landscape. And the core strategy that we're using for this is based on a network, excuse me, of nurse sites. So these would be locations that have robust populations of the agents that can facilitate spread to other locations. So if we imagine some populations here in Southern Ontario that are maybe a few years old and doing really well, we can expect spread from that to happen naturally. These are living organisms that are looking for new habitats. They are moths. We've observed flight up to 2 kilometers within a year, sort of as our high water mark so far in terms of dispersal capacity. So they will spread out on their own over time, which is great. We can also look at ways to assist their redistribution over larger distances or to specific locations, site to site, to avoid having to go back into the lab and go through the whole rearing process. So we've been looking at some tools to do that as well. We mentioned the eggs are laid on the leaf sheaths, sort of up against the stem. So they're there by the end of the summer. So what we've learned is we can go in, we can look for where those eggs are. They tend to be on the lower part of the stem, so most will be 1 meter or lower. So we can harvest stems from an established nurse population, lop off sort of above that first meter or so. And even if we don't dissect them, we can predict a certain amount of eggs will be on those stems. Those stems can then be moved directly to an additional site. And this stem transfer technique we've just started piloting it this year, but so far we've been seeing really good results with that so far. So we're hoping to use this even further starting this spring as a new release method for moving agents site to site. So how are things looking so far in terms of releases and long-term monitoring? I pulled my title slide from the 2022 sort of talk there. So in some ways, some things don't change. Apparently, I turned this photo 90 degrees, sort of one of the main differences there. But in terms of the program, actually quite a bit is different. So this is what I was presenting at the time. We had 8 sites as of that presentation, just shy of 9,000 insects, 8,500 there, distributed across those sites. Currently, before sort of going into this spring, we have 50 sites distributed across Southern Ontario, stretching from close to Windsor all the way up to North Bay and then just east of Cornwall and Ottawa there. And we've released approximately 30,000 insects from both of those species. So we've been able to put out a very consistent and growing amount of material every year, which has been great. There's a lot of information here, but those are those 50 sites from 2019 to 2024. And basically what we're looking at here is a high-level overview of the monitoring data that we have from these sites. So we monitor all of our sites on an annual basis. What we're looking at is, if we look at that year one in blue column, this is the number of damaged stems that we find per meter squared, so damage density, just around the plot. So we put out some larvae, we look at how much damage is around that plot. In green in the subsequent columns, this would be also damage per meter squared, but this is measured at a whole population patch level. So this is that follow-up monitoring where we're looking at an entire Phragmites population in subsequent years. So we now have this growing inventory database that we're going to be adding to every year as we release new sites and go back to monitor old sites. And we can pull out a few key patterns from this to make it digestible. The first is that releases are going quite well. So 93% of the releases that we're able to sort of measure in that first year produced damage. And those that don't, we usually know why. Sometimes it was, excuse me, a very marginal site, or it was done at a suboptimal time in the season. There are very few that failed for reasons that kind of are unknown to us. So generally with the right timing and conditions, releases go well. To give you an idea of what that looks like, this is from our Long Point site, one of the sites in the Long Point area. You can see a piece of white PVC there. That would have been a release that we did early in the spring of that year. You can see there's a hula hoop there for counting stems. The damage is relatively obvious in person, but hard to capture in a photograph. But you can see here, each of the stems with a blue flag on it has been fed on by the biocontrol agent. So we have a nice amount of damage starting to spread away from the release point. This is exactly what we like to see, and that's something that we can quantify at each of our release sites. The second thing we can see is that damage generally persists and increases over time. So when we have damage in one year, we go back the next year, we continue to see damage. And generally, it tends to increase sort of year over year at many of these sites, or at minimum, remain relatively consistent. So again, we can sort of go back in, except now we're focusing on monitoring a much larger area, and we're looking for damage anywhere in that patch that they might have dispersed to. When we see damage in year 2 onwards, this is particularly encouraging because those are the offspring of insects that we released. So it means that they are reproducing, they're overwintering, and they're spreading at the site, which is great. Another important takeaway is that we don't want to give up on sites right away. We've had a few situations where we release early on and, excuse me, basically don't see anything or see very low levels of damage. But when we come back in subsequent years, 1, 2, 3 years on, usually damage does show up. So it's a reminder that, again, this is a sort of long-term strategy here. And it's really important to be monitoring these things over multiple years because a very small, initially undetectable population can eventually become quite sizable and impactful. And so finally, what we've been focusing on so far has been monitoring the insect populations themselves by looking at damage. But of course, what we're trying to do here is try and manage Phragmites. So increasingly, now that we have more sites that are sort of moving towards establishment, we do want to focus more on monitoring actual impact on Phragmites and vegetation community overall. So we've started to develop some protocols for this. Generally, this happens in the fall towards at the end of the growing season, and we can go in and measure things like the density of Phragmites stems, their height, how many are forming panicles, and that sort of thing. We do have data on this that generally shows you get reductions in these. So when you have feeding and you have stem mortality and reduced sort of dominance, we see reductions in these things that can be quite sizable. But we are cautious with this because it is very early plot level data. So generally this is coming from a fairly constrained area immediately around where we release. What this looks like at a population scale, at a patch scale over multiple years remains to be seen, and that's what we're monitoring and will sort of really be the main story to tell. So this is a very, very encouraging start, and we're hoping to see this pattern replicated over time at much larger scales, which would be very, very meaningful in terms of reducing Phragmites dominance making space for native vegetation and so on. So what's coming up with the program and long-term program goals? This is an operational program where we are trying to get insects onto the landscape to help manage Phragmites. But this is, of course, supported by very essential long-term monitoring and research efforts to understand what's happening and to improve the efficacy of the tools that we're using. So just a few examples of some of the work that's been going on recently. I mentioned we did a pilot study to trial this stem transfer technique. So you can see there in sort of the bottom middle, those are bundles of 75 cut stems that have been moved from one site to a new site. We were experimenting, excuse me, a little bit with when you cut the stems and when you move them into a new site. And generally it was fairly agnostic in terms of seasonality. So you could do this at different times of the year, and it was very, very effective. We do have two agents as well. So one of our big questions recently has been, is it better to release one species or two species at a site at the same time? So we had a 12-site release experiment, so a fairly large investment of effort in 2024. 1/3 of the sites got one species or the other or both together. This is a long-term study, so we have some initial data that suggests at minimum combined releases are probably just as good as single species, maybe a little bit better because of some, excuse me, complementarity in terms of how and when they feed, although it's a little bit early to say. So an encouraging result so far, and we'll continue to monitor this in the years to come. Another thing, very operational question we were looking at is whether we can control the hatch timing of insects. So usually they're hatching sometime in late April, early May, if they're just kept under outdoor conditions. But we want to see if we could control that timing a little bit to excuse me, make that release season a little bit more manageable and to synchronize the agents with the phenological status of Phragmites in the field. So make sure the larvae are encountering frag that's sort of the right size and the right density to feed on. So we did a cold storage experiment where we were taking eggs and putting them into the cold, basically in the fridge, for different periods of time. And we found that we were able to delay hatching by up to a month. So that really gives us a little bit of wiggle room and flexibility in that timing without any real sort of detrimental impacts on the insects themselves. So that's a new tool we're going to be using as well. So in addition to those examples of some of the research that's happening, again, this is really about getting insects onto the landscape. So our goals going forward as we're in this integration and scaling up phase is to grow the network that we have of nurse sites that will allow for that natural and assisted dispersal and redistribution. And increasingly, we want to look at how you actually integrate biocontrol as a tool with other management options. So again, we're adding a tool to the toolbox, not throwing the toolbox out the window and introducing this single silver bullet kind of solution. So we really want to know, how do those tools work together then? So some of the questions we might want to ask, for example, is, one of the main challenges is that you can spray or you can burn or you can mow or whatever you're doing, but you're going to get regrowth. So how effective is biocontrol at suppressing, slowing, or limiting regrowth that happens after other forms of management? So if you have a managed landscape with biocontrol nearby, how much does that help? We also know that there can be very practical constraints on ability to spray or burn different types of sites depending on sort of the geography of the landscape or built environment and so on. Spatially, one other big question would be, can biocontrol be useful as a very mobile, flexible tool to effectively mop up some of those more isolated, hard to reach islands of frag that either couldn't be treated in the first place or are regenerating, but in sort of small clusters that are just not really feasible to efficiently manage with other tools. We also know, of course, that with many weed management programs, restoration is really an essential sort of piece of follow-up here. So can biocontrol facilitate native plant recovery following restoration? What does management look like when we are removing Phragmites, actively restoring or fostering regeneration from a seed bank, let's say? How does biocontrol help that process, ideally by sort of tipping that competitive balance between the different species in that vegetation community? So to summarize some of these key ideas here, from 2019 to present, we've developed now these core operational protocols for the program, how we rear them, release insects onto the landscape, and monitor their performance distribution, and increasingly we're looking at impacts as well. So to date, these tools have been used to release approximately 30,000 insects across 50 sites in Ontario, Canada at this point. The monitoring data that we have to date, and we're building on that database year after year, tells a very encouraging story, and that suggests populations are beginning to establish on the landscape. So generally we're releasing once and we don't go back to introduce more insects. That's a population that is sustaining itself. They're beginning to spread. We often see patches nearby that we're monitoring and keeping an eye on that we didn't release into, that become colonized within a couple of years. So again, that's exactly sort of what we want to be seeing with that, and also having an impact. So we know on a plot scale, we see reductions in Phragmites dominance, height, density, panicle formation, and we're going to continue monitoring to see how those effects scale up at more of a population patch level as well. As the program ages, we're shifting our focus to, again, more of the scaling up and integration of biocontrol with other tools in the toolbox. What does that look like before other management, after other management, alongside other management? That's sort of a major area of inquiry for us this coming spring as well. Of course, with this audience, so there are no US releases yet, but the petition remains under review. This is an ongoing international collaboration, and it always has been. We continue to chat with colleagues in the States who are supporting this work as well. And there is natural dispersal as well. So as we said, these are moths and invasive species, and also biocontrol agents tend not to respect sort of international or interprovincial or interstate borders as well. So there is that potential, expected potential for natural dispersal in time as well. So they will get there regardless. In terms of additional sources, just some of our sort of published work on this, if you're interested in pursuing some of that as well. We have an open access chapter in sort of CABI's most recent book about biological control programs in Canada. This is available online and open access. Provides a really nice overview of the Phragmites system, but also the history of the biological control program and an update as of 2023. We did some work with another postdoc in the lab, Rhoda de Jong, early on, one of the first projects we worked on, looking at characterizing overall insect communities that form on native and introduced subspecies of Phragmites australis. So we haven't talked a lot about native Phragmites as well, but it is a major focus for us as well. It's important to note that both of these species, subspecies, have fairly diverse insect communities associated with them. And understanding how those change in response to biocontrol agents being added to the mix is something we're monitoring as well. We have a very sort of applied tool that we developed as well for helping identify between native and introduced subspecies of Phragmites based on morphological traits. So of course, it's important to be able to tell what you're looking at and ensure that you're controlling the right subspecies here. Genetic testing is, of course, extremely useful, but sometimes hard to access in some cases. So we did a survey of several dozen sites, native and introduced, throughout Ontario and around the Great Lakes region. And we identified statistically a suite of 5 morphological traits that provided rapid, simple, reliable separation of the subspecies. So this is available in that article as well. Designed for, again, sort of more of a Southern Ontario habitat, but also Great Lakes, so likely applicable to a broader region as well. So I'd encourage you to check that out if you're trying to figure out which subspecies you're looking at. And then we have a couple of recent publications talking about the biocontrol program specifically, looking at initial testing of some of the egg and larval release methods that we trialed in the initial years before we arrived at the tools that we are now using. This last one here in +1 was sort of a broad overview of the biological control program as of that year, with some preliminary analyses of the monitoring data that exists to date. So with that, I'd like to say again, thank you very much for the invitation to be here again. I'm very happy to entertain questions here. There often are a lot, and always happy to discuss that. And if there's anything that we don't get to today, please feel free to reach out and email us here. My contact info is there. I should also flag that I'm actually going to be transitioning out of this role sort of in mid-April. So don't worry, the program will be in very good hands. In particular, my colleague, Ian Jones, who's also a research associate in the lab and sort of my partner on this so far, he's going to be probably the lead on this program going ahead. So if you have inquiries after sort of into April and onwards, I would suggest directing those primarily to Ian there, and he'd be very happy to help you. So with that, thank you, and happy to take some questions here.
Thank you. That was fantastic. I think, hold on, I think I have like a little, unfortunately it doesn't have your email address. So I guess if you wanted to put your or Ian's email address in the chat function,
yeah,
that could always be helpful. If you don't mind making it public, if you don't want that,
no,
that's also
no, for
so
sure,
fair.
of course. Putting that in there right now. Yeah. There we go. Okay. Yeah, that's both of us.
Yeah, so like I said, feel free to put any questions you have. You can do the chat or Q&A. I'm reading off the Q&A, but Nicole, my colleague, is moving everything in the chat to the Q&A, so hopefully I don't miss anything. It does make it hard that there's like two functions. And so I'm just gonna kind of start off, and we can hopefully get in however many we can get in like 17 minutes.
For sure.
So from Angela, how far away from the release sites have you recorded the moth spreading?
Yeah, so our sort of high water mark for this, the maximum dispersal we've noticed in 1 year so far has been 2 kilometers. I suspect that's going to be on the upper range of what is typical. So you can have very random events where a moth sort of flies up and gets caught on the wind and gets pushed somewhere and manages to find another population. So I think the average will be lower than that, but we've seen up to 2 kilometers. And we're trying to learn more about what that dispersal capability looks like right now with pheromone monitoring of the moths in particular to understand how they spread. So, very short hops, quite easy within a few hundred meters, up to 2 kilometers has been seen so far.
That's crazy. And then also from Angela, I'm guessing not you particularly, but this program, have you engaged with any First Nations about the release sites? And if not, are you working with them on a process to be involved?
Yeah, definitely. So whenever we do a release, wherever that is, that starts with a conversation with whoever those particular land managers would be, which includes First Nations in many cases and other sort of partners. Because of the somewhat irreversible nature of biological control, of course, it puts a real onus, I think, on those kind of discussions. So that happens during the petitioning process. Broadly, these kinds of conversations with all sorts of stakeholders, but we also conduct these on a case-by-case basis as well. Whenever we're sort of doing a release in a particular area, that always starts with a discussion. So these are sort of allowed to be released anywhere in Canada, but we won't do that without sort of land manager consent and discussion, of course.
Thank you for elaborating on that. Has there been any evidence that the moths are eating like native species, native insects?
So certainly, no, certainly not insects. I mean, these are herbivores.
Or so other species of plants, I should say.
Other plants, yeah. So no, that 20-year period prior to the petition is mostly dedicated to that host specificity testing. So these are species that have a very long, deep coevolutionary history with Phragmites. They are specialists that feed on Phragmites australis only, feed on and develop successfully only on Phragmites australis. And that includes subspecies level specificity as well. So they will choose introduce Phragmites, invasive Phragmites over the native subspecies as well. So that specificity has to be demonstrated before the petition is approved. And that has been consistent with what we've seen in the field as well.
Awesome, thank you. I guess, and this is from Lenka, in light of many years of independent research into the impacts of glyphosate on non-target species, do you think biocontrol will be expedited in any way as like a management control tool?
Yeah, that's a very good question, Lenka, and also I saw your email to Sandy and Ian as well to our group. So yeah, thank you for reaching out, and I read the article that you'd shared there. That was very interesting. I'm not an expert on what's going on with the sort of glyphosate regulation right now, but it was interesting to read that. That certainly could be factored in. I know petitioning processes are generally focused on risk of the specific tool being considered, and the US process in particular has that primary focus. The Canadian process, one of the differences is it does factor in a little bit more of the relative risks of other tools and also the benefits associated with implementation. So that would factor in something like, if we have less reliance on glyphosate or other chemical control, there's a greater need for additional tools. I'm not as familiar with the US system. I think it doesn't weight it as heavily, although of course that's still going to factor into the needs of land managers and how people think about this broadly. So one would hope, I think.
Yeah. Yeah, I don't know about it too much from the US. We know a little bit, we've heard some updates, but yeah, I agree. I don't think that it's more like tool related versus.
Yeah.
And then you kind of addressed this in actually 2 questions ago, but just to clarify for Matthew that as I understand, these moths do not affect agricultural crops such as corn and soybeans, and so no.
Yeah, that specificity has been shown on, and agricultural and sort of economically significant crops are sort of key part of that testing as well. And there's a variety of reasons for why that happens. They need sort of specific stems to bore into because that's where they feed. It's also where they pupate. They also need standing vegetation that will retain its leaf sheaths as well, because that is where they oviposit. So anything that sort of has complete senescence and doesn't retain that structure also just wouldn't be a place where they could lay their eggs and overwinter. So that's one of the reasons we see the preference for introduced over native Phragmites as well, is that native Phragmites tends to drop its leaf sheaths in the fall and in the winter. So even if any eggs were laid on that, those would drop off and be predated upon or desiccate or something like that, because they don't have that protected sort of overwintering site anymore.
Yeah, thank you. And I think this question kind of will follow up on that response. So that from Angela, is monitoring being conducted on non-target plant species? And so I guess you could include like native Phragmites.
Yeah, so the main one we are monitoring for that because there's a very high level of confidence in that specificity. The one that we are still watching is native Phragmites. So we try and prioritize, for example, release sites that have on the landscape fairly close by introduced Phragmites and also native Phrag. And we have several of those sites so far, and we monitor them every year, and we're not seeing any feeding in those areas. The other thing is that there's a difference between a moth laying a few eggs and a caterpillar eating a few stems, and a population successfully establishing, growing, and having a population level impact. We definitely don't see the latter, and we don't even see the former, even for a very taxonomically similar sort of subspecies, which is really good.
Yeah. Another question about concerns on native Phragmites. I love this, but yes, you kind of just addressed it, that this is something that's being monitored. You guys have so many questions. This is fantastic.
Oh, it's great. I can amend a little bit, not amend, but an addition to that, I guess, as well for native Phrag is really the major threat right now to native Phrag is invasive Phrag. We have a hard time finding it because it sort of has been swallowed up by invasive Phrag generally. So even if we saw any feeding or sort of population level impact which we don't, and we've been monitoring for and we don't see it, that would still likely be sort of lower than the impact that we see all the time from invasive Phrag as well. So sort of the cost benefit analysis there would still favor even a tool that wasn't quite as specific. But so far we haven't seen any of that anyway, which makes it sort of definitely the right tool to be using there.
Yeah. Is this program looking into additional release sites in like the Toronto area, or I guess in general in Ontario?
Yep. So every year we are doing new releases. Generally, these are planned at least a year out. So we have sort of a fixed amount of rearing effort and capacity that's usually based on how many insects we have to release. We've sort of realized in the last couple of years that you can have a successful release with a very small initial number of insects. So even a couple hundred is enough. but they tend to grow, as you might expect, very, very slowly. So we're seeing sort of better bang for the buck if you do a larger, more concentrated release. That population will get off to a better start faster. So we're doing a little bit more of a concerted effort at a smaller number of sites each year. So our spring 2025 sites are basically planned out at this point. They're focused mostly for those sort of in Southern Ontario, familiar with the area, more in kind of the Durham region. We're working within some of our provincial management frameworks for that. So focusing on a particular area, plans for 2026, sort of, we'll start looking at that as soon as now. So we're always happy to have conversations with people about future releases.
Awesome. Oh, we just a question, and you might not know this, and we could probably help address it, but what agency is in charge of permitting on the US side that could be encouraged to expedite the permitting process? And I believe that'd be USDA APHIS.
USDA APHIS is sort of, that's right, yeah. So I know at this point in the process, so the so TAG sort of has sort of recommended it for release, and then I guess it's under review by USDA APHIS at this point. But again, I'm not an expert on the US process.
Yeah, same. And I think we tried to get an update. I know one of our of where they were, and not super responsive at the moment. So if we get one as the Phragmites Collaborative, we'll let you know. But yeah, it's a very long process on the US side. So unfortunately. Yeah. Any other questions, Nicole? I know you've been adding them so I can see.
I'm not seeing any at the moment. Just some more information on glyphosate from Lenisa, if you want to check that out, Michael, in the Q&A. But
yeah, that's great. Thank you.
Not any super direct questions I'm not seeing, but that's the only one that I'm seeing we haven't answered yet.
I can't type anything in the chat.
Oh, interesting, Emily, but you can unmute, so feel free to ask your question.
Well, that's why I was raising my hand. And maybe you would already answer that. I was kind of wondering whether the complications when it comes to relations between U.S. and Canada might affect the furthering of this study.
Yeah, thank you, Emily. That's, of course, a concern across many avenues of environmental research work right now. I would say, again, we have colleagues and partners sort of in the US at Cornell and sort of others, and we're continuing on with the work that we're doing, sort of unaltered by things. At the end of the day, the researchers who are doing the research, they just want to get that done and understand that these are cross-border sort of issues. So until there is any kind of specific legislation or other sort of barriers that get in the way of that, and on at least a individual researcher level, it's kind of business as usual, sort of in our experience for the most part. But that is definitely is a concern for people. It's on our minds for sure.
And we do have one more question I saw popped up from Arlo. Has there been any plans for testing this biocontrol for Phragmites in more saline environments?
Yeah, it's so definitely sort of variety of site conditions, which would include hydrology, salinity, are of interest. I think we're kind of entering the stage of the program where we can start to look at some of those things. For example, we started with a lot of dry sites just because they're easier sort of to work at, and then started to work in weather and found that they work just fine there. One potential area we're looking at in the coming years as well is sort of contaminated sort of industrial sites as well, where you have metal toxicity and things like that might be a concern. So certainly all of these, including salinity, would be things we want to start looking at in the future. And some of this will happen naturally as well, in that the moths will be dispersing where they want to and sort of avoiding sites they don't want to as well. So that can tell us a lot as well, in addition to more targeted releases and experiments that we do ourselves actively.
That's awesome. That's really, really interesting. And not a question, but a comment, I think, in response to our previous question from Emily. Austin just noted that they're on the team with the US collaborators at Cornell and are still going forward with the collaboration, and nothing has slowed us down yet. So that's awesome to hear. So thank you for chiming that in, Austin.
That's great. That's a sentiment on our side as well.
Okay, unless I missed anything, Nicole can let me know. I think we have all the questions, which is great. Thank you for leaving so much time. I knew there was going to be a lot. So,
For sure.
and then I think I just have one. So after this is done, in the next day or so, you'll get an email from Microsoft Teams. It's like the recording is ready. So feel free to share that if you have colleagues who want this information and weren't able to come today. You can share the recording. It'll also be uploaded to our YouTube and website as well. But you should get an email that's ready to view. And then just a plug for our April webinar. Registration is now open, and we'll also be talking about more really cool up and coming Phragmites research